This is Bob and Sue Anderson. They are expecting an addition to their family. They have decided to see a geneticist to see about any potential diseases their baby could have. The test results come back for Bob and Sue, and they learn that they each have a mild type of thalassemia. Thalassemia is a genetic disease affecting the protein hemoglobin, which is made up of 4 subunits, 2 alphas and 2 betas. If there are any mutations in the genes coding for these subunits, it can lead to many different health problems such as anemia, enlarged spleen, and/or bone deformities.
Sue has alpha thalassemia. It is named this because there is a mutation in one or both of the genes encoding for the alpha subunits. These 2 genes are HBA1 and HBA2. There are 4 different types of alpha thalassemia. The 4 different types have increasing severity of symptoms as more alleles are mutated. There is a wide variation of symptoms from no symptoms for silent carrier to fatal for major.
The other kind of thalassemia is called beta thalassemia. Bob was diagnosed with this type. This disease occurs when there is a mutation in the gene called HBB, which codes for the beta subunits. There are 3 severities of this disease. The mutations for the HBB alleles can either be a partial loss of function or a null mutation. The severity of symptoms increases similarly to that of alpha thalassemia. Beta thalassemia minor can have no symptoms or mild anemia, while individuals with major require regular blood transfusions to survive.
Statistics for how prevalent this disease is worldwide are usually underestimates because of its tendency to be found in underdeveloped regions where testing and treatment is deficient. Alpha thalassemia is most commonly in Africa and Asian countries, while beta is more commonly found in the Mediterranean. Their geneticist, Dr. Martin, decides to use the CRISPR-Cas system to quickly make double mutant mice to study thalassemia. CRISPR is an RNA molecule that Dr. Martin will synthesize to pair with all 3 genes involved. The pairing of CRISPR will recruit Cas, an endonuclease, to cut out the gene sequence. This is a representation of what the CRISPR-Cas mechanism looks like. The guide RNA in the picture is the RNA that recruits the Cas endonuclease, which is shown in green.
The double mutant mice that will be generated will tell Dr. Martin if individuals with mutations in both alpha and beta genes are viable. He would look for the common associated health problems in his mice in order to determine the severity of their phenotype. Further experimentation would need to be performed in order to look at all possible genotypes, including the partial loss of function mutations present in the HBB gene. These are the possible double mutant combinations that could be generated from the experiment. There are 3 possibilities for the interaction between the alpha and beta genes. They could not interact at all and only exhibit their individual phenotypes. They could have a positive interaction where the phenotype worsens, or there could be a negative interaction which would result in a less severe phenotype. Studying the phenotypes of the mice from this experiment would help Dr. Martin predict the phenotype of the Andersons' child.